Okay, so we will see in more detail how it is defined. And then the fourth parameter is polarization, which is not something that I'm going to discuss in detail, but this is again something which makes sense in the excited state. Like suppose polarization is basically we're talking about the orientation of the dipole. Suppose you have a molecule, so you have like the dipole suppose in this direction. Now when it is excited there in the excited state, whether it remains like this or it makes a movement, it tumbles down. So that is basically the orientation of the dipole, how it changes in the excited state, and whether that can be used in some sort of extracting information from the system. So this is a very simple slide of telling what is fluorescence. So shine light in, get absorbed, re-emit at a longer wavelength. This is precisely what I just said in the very first slide when I was trying to explain Jablonski diagram. So you have a molecule which gets excited by absorbing a photon, then it comes back to its lower vibrational state. As I said, this is has to be v equal to 0 state. In the process it emits some energy, and this difference in energy is nothing but the difference in absorption and emission photon. So Jablonski diagram has told, so I will not get into more detail, but throughout my discussion of whatever I'm going to talk about is mostly associated with fluorescence spectroscopy for the reason that it is very sensitive form of spectroscopy. Then coming to lifetime, as I said, like you excite the molecule, so how long the molecule stays in the excited state. So if you can actually find out the population of molecules in the excited state and how that changes with time, so this is a plot of logarithm versus time. So ideally excited states they decay exponentially with time. So you can tell that suppose if you have a emission intense, if you have number of molecules in the excited state at some point of time, let's say t, and then you need to know how many molecules were there in the zero time, and then it exponentially decays. Now when you plot logarithm, then it actually comes as a straight line. So these are again part of measurements, that how we measure it. So the typical instrument that is used for measuring excited state lifetime we call time correlated single photon counting. So with that actually you can mostly molecules in the which shows lifetime in the range of, let's say 0.2 nanosecond, I would say, on the lower side, and then you can go as long as 100 200 nanosecond. So you can measure molecules which displays excited state lifetime in this regime can be measured with TCSPC spectrometer. Once again, just to emphasize how lifetime is, what is the meaning of lifetime. So the molecule stays in the excited state for an average time. So that is the average time that molecule stays in the excited state is the excited state lifetime. Now I have just given a pictorial presentation with respect to a different analyte. So there are many variables. So the whole idea about absorption and emission spectroscopy is that you should have a molecule which you are trying to investigate. Now that molecule can be anything. It can be an organic dye molecule, it can be some inorganic materials also. But the idea is that it should absorb the light in the visible region, because when we talk about absorption spectroscopy and emission spectroscopy, we tentatively assume that this spectroscopy is dealing with somewhere, let's say 190 or 200 nanometer to up to around, let us say 900 or 1000 nanometers. So that's our visible region, and your molecules should be able to absorb and emit light in this particular wavelength region. Now when a molecule absorbs the light, so then what happens, it will have a peak position, we say absorption maxima. Okay, so that is one information that we read out of the molecule. Another is what, okay, absorbance, we say optical density, right? All of you are aware of it. So optical density, so with that optical density we can find out the concentration of the molecule in the solution. Now when we measure the interaction of that molecule with, let us say, some another analyte. So I have a molecule, let's say A in the solution, and then I have put B into it. Now B is spectroscopically silent. When I say spectroscopically silent means it does not have its own absorption or emission features, but then I believe that this molecule A, which has its optical property in the form of absorption and emission, will interact with B, and in that process it will give some changes, some features, some signatures in its absorption or emission spectra. So there are many parameters that we read out of absorption and emission spectra, like peak position, intensity, optical density, lifetime, anisotropy. So these are some of the things that I have listed here. For example, this is an emission spectra. So suppose with a variation, with some interaction with a particular, light, let's say analyte, the peak position varies. So what does it indicate? So basically intensity and spectral position. So these are two important observables from emission spectra, and the same applies to absorption spectra also. Then let's say lifetimes, there are some 4-5 decay traces. So suppose if a molecule is free form, and so there are, can you see the rate graph here, somewhere here, cursor is okay. So so basically you see the molecule decays very fast. Very fast means the intensity is decaying with time at a very faster rate. Now if you look at the pink graph, then this also decays, but it decays down with a slower rate. See, the idea is every molecule has to come down to the ground state, because a molecule does not like to stay in the excited state for a longer time, for the obvious reason that it is rich in energy, and every molecule wants to get to its ground state with time. So, but then there are some phenomena that can happen in the solution which will allow the molecule to stay in the excited state for a longer time. So these are, I'm talking about, these are different observables with which we can actually do the molecule, or whether the molecule is feeling a very different environment. Suppose if I pull in the form of decays very fast, and then I put B into it, and then that the line has been, because it remains in the excited state for a longer time. So that way we can say that the lifetime has increased. So these are various parameters, again, as I said, like in terms of anisotropy also. So my idea is just to tell you that there are different experimental parameters that we try to read out of these spectroscopy, both absorption and emission, and they provide us different kind of information. And when I say different kind of information, we will have to take specific examples, and then we will try to understand at a molecular level that okay, this is happening, and that is why there is a change in the, let's say, peak position, or maybe let's say there is a change in the emission intensity, or let's say lifetime, and things like that. And from there we extract the information about what is happening in the solution. Okay, so these are like typical example of information that we can obtain from these kind of things. Is like, let us say, a kinetic parameter we can measure, and then what are the properties of surrounding environment, then various structural details and binding features. So these are all basically explained in terms of a fibril, a dye when it is put in a fibril solution. So I think if I have got a chance, I will get to it. But still, since I'm discussing, I must tell what is a fibril. So fibril is nothing but some sort of protein aggregates. Protein normally, when it is in the native form, when we say native form, basically, I mean, all of you are aware the proteins have its native structure in the form of secondary and tertiary and quaternary structure, right? So when it is in the native form, it has its best function, and that is when the body preserves it. But for many reasons, the protein may get unfolded, and when it unfolds, then it starts behaving in a different way. And when these unfolded proteins start aggregating together, so there is a particular structure that is called amyloid fibrils. So these kind of fibrillar aggregates were found to be formed in Alzheimer's disease. So that is how there is a lot of interest in this area of studying protein aggregates and how they behave, how they change with the different kind of proteins, and whether you can identify them with some kind of absorption and emission spectroscopy. So I have a dedicated presentation on this on a different note, but since I have put up this slide here, so I just thought maybe I should tell. So the idea is that whatever is there in the solution, whatever medium you try to measure, you name me anything, let's say a protein or a DNA or a fibril or heparin or any other analyte that you want to sense, you would like to choose a molecule which has optical properties, like in terms of absorption and emission features, and you would choose the molecule in such a way that when it comes in contact with that particular analyte, then you will see the changes in its spectroscopic properties. When I say the spectroscopic properties, I am focusing on absorption and emission features, and when I am talking about absorption and emission features, I'm going to read many parameters from it. Let's say the intensity of the emission, peak position of the emission spectra, lifetime of the molecule, absorption peak position, and things like that. And from there, I try to get information. Okay, my molecule is interacting with this particular analyte in this particular way, and that is more on the fundamental side, that okay, you know that okay, this is how it is interacting. But the more important question comes, that whether this information that you are trying to get out of these spectroscopies is of any use to the societal requirement, right? So that is where my talk is focused at. So we will see that how these particular features that we are trying to read out of the system is not just of an academic interest. It is not just some spectral changes, some spectral peak positions happening, but we try to translate into the form of a sensor. When I say sensor, okay, so I have a molecule which I want to detect, but then I need to be very specific that okay, I choose a molecule and this is my analyte of interest, and when it interacts with it, it gives a specific signal for this particular molecule only. If I put any other molecule and it also gives a similar feature, then it is of no use. It only tells that my molecule, which I say my probe molecule or the optical probe molecule, is interacting with it. And maybe I can tell okay, this is interacting in this way. Let's say it makes hydrogen bond, let's say it interacts like electrostatically and things like that. So on the fundamental side, but then whether that information is useful, can we really utilize that information to do something, right? So with that, let me just tell you that these are some of the my sensor activity that I have been doing in last 4 or 5 years, I would say, to be precise. So the variety of analytes, like for example, say human serum albumin. See, human serum albumin is most abundant protein in the body, and its levels in the human serum is associated with several diseases, especially related to liver and kidney functioning. Especially, let us say in urine, there is something called microalbuminuria. So in the early phases of this, which is related to basically sort of a kidney disease. So if a small amount of serum comes or start coming out in the urine, it is an early sign of a very severe kidney disease. So then you need to basically have a molecule which can detect a very small amount of albumin in the urine matrix. Now we look at the challenges in this. So first of all, along with albumin, something else can come. So your molecule should be able to only see the albumin in the urine. Now urine has its own matrix, it has many other components, salts and many other things. So your probe, the probe that you are choosing, should not be basically showing some signal with respect to that matrix. So in this particular work, basically we have made a device basically which can detect albumin in the serum matrix. So I think if I get a time, I'll get you like which molecule you should choose, what parameter you are looking at, and how you kind of try to devise a sensor in it. You cannot pick up any combination. You should have certain combination, and then many other factors will come into play when when you go ahead on the journey. So let me see, I will share my journey at least with respect to one analyte, but I've listed few analytes just to give you a glimpse of what kind of various analytes we can sense, or at least I have attempted in last 4 or 5 years. So this was on serum albumin. Then there is a per-renate anion. Per-renate anion is basically it's a non-radioactive surrogate of hazardous per-tignate anion, which is a concern in the nuclear waste, and which has a very large half-life. And when a small amount of it gets into the environment, it could be a big concern. So by the time I got into this area, I actually this was the first work which actually spoke about that there is a fluorescence way of sensing these analytes. By the way, I just want to know if my speed is fine. I'm not getting very fast.
It is fine, sir.
Okay, great. Okay,
A
so
bit
now
slow
coming
would
to
be better, but it is also fine.
okay, no issues. I think I will try. And then in this work, we try to identify or distinguish between metalloproteins and non-metalloproteins. So a metalloprotein and non-metalloprotein, as the name says, basically it is distinguished by the presence of a metal. Now we had a supramolecular assembly which actually had a very different kind of response towards these two different class of proteins. So So suppose this is my probe, I would say, let's say emission is coming out of it. When I put a metalloprotein, I see a turn off response. What is turn off? So turn off is, suppose your molecule has some emission in the free form, nothing has been added to it, right? Now I add one analyte to it, and upon the interaction with the analyte, the emission gets quenched. This word you must have heard, it gets quenched. So that is, we say, is a turn off response. Now suppose if it is the reverse one, that suppose if I add analyte to that particular probe molecule, see this, I am going to use this term in the terms of, let's say, emissive molecule, a probe molecule, a reporter molecule. So the idea is I should have a molecule which is my reporter. Reporter means that will tell what is happening. That will tell what is happening in its surrounding, in the system. So that is my reporter or probe or molecular probe, and which should necessarily have absorption and emission features. Now only absorption and emission features are not good enough, it should have a sensitive feature. So you can, it's not like you take any fluorescent molecule, you put it in the solvent and add your protein and see a change. No, it doesn't happen that way. So, and that is where the choice of molecule comes into play. So you have to choose a molecule which will, upon interaction, shows a large difference. So those kind of molecular choice has to be selected when you are choosing a particular analyte to detect. And so basically, very interestingly, we saw that in a special class of protein, let's say metalloprotein, we see a quenching, whereas in another class of protein which is devoid of metals, we see a turn on response. And then similarly, as I said in one of my slide, that amyloid fibrils. So amyloid fibrils, as I said, these are basically protein aggregates which are toxic to the body, and these are found to be abundant in patients which suffer from Alzheimer's and Parkinson's disease. And of course, I am not the first one to deal with these kind of molecules. This research have been going on for decades, both India and abroad. I think when I was in US during 2000, as I said in the beginning, that we have a spectrum from, let's say, we are talking about 200 to, let's say, 900, 1000 nanometers that we consider, let's say, this is a visible region for us, UV visible, and let's say NIR. So that is the reason in which our molecules are focused. And now suppose if your molecule needs to be excited at 300 nanometer, you came up with the molecule and you tried to excite it at 300. You said that okay, my absorption maxima is 300 nanometers, I have to excite at 300 nanometer. Now what is the problem with this lower wavelength excitation? So when I talk about biomolecules or biospecies to detect, then you will have to understand that there is a matrix. Like you want to detect something in the blood. Okay, blood, you get red blood particles, you can get it, but then you have a serum. Serum has multiple proteins and multiple species which absorbs in the blue light. For example, proteins, tyrosine, tryptophan, these are amino acids which absorbs in the blue region and emits in the blue region. But then I want to detect something else, but my background absorbs that light. So there is a standard notion that if your molecule, if you can excite more on the redder side, red side means, let's say, go anything more on the higher wavelength. See, a 500 nanometer is a redder wavelength than a 400 nanometer, or a 600 nanometer is a redder wavelength compared to 500 nanometer. So things like that. So people try to define, make new molecules which can absorb longer wavelength of light and then give absorption and emission feature. Why this all business in shifting the absorption maxima to a new domain? It is basically nothing but to avoid autofluorescence. As I said, that the matrix itself has many different kind of species which absorbs light and gives emission. So we actually could find out something in the near infrared. So near infrared means it has less problem from the autofluorescence. So I am trying to excite my molecule with a wavelength which kind of evades or avoids exciting various background species in the system. So okay, just a minute. Yeah, so I think a couple of more things were there, but maybe if I get time later on, I'll tell you. But so this is something that I wanted to discuss because I want to make a generalized story so that you understand the importance of one particular, let us say, spectroscopy, and how in tackling one particular problem, how step by step we have gone ahead, right? And from where the journey. So it's a complete story, makes more sense, because in the previous slide I was trying to give you a glimpse that what variety of analytes we can sense, and then of course it has to be something very important. I mean, you would not like to, develop a sensor for, let's say, magnesium or mercury or things like that, for which things are already developed. So I have to target the important analyte and then accordingly think about, what probe you can choose. So my discussion, majority of the discussion, I would rather say, for today's talk would be mostly on sensing heparin. So let me first briefly tell you why heparin. So heparin is basically the second most widely used natural drug worldwide after insulin. Insulin, you all know, diabetes management and things like that. So heparin is the second most widely used natural drug. And the reason is that it is widely used as a blood anticoagulant to prevent thrombosis during surgery. So when surgical operations goes on, so the blood needs to flow, otherwise the blood will clot. So you have to keep them in the flow. So anticoagulant needs to be added. So heparin is one of the most widely used blood anticoagulant, and it is also used as a therapy for thromboembolism, deep vein thrombosis. These words you might have heard during COVID times, that especially in the second wave, you must have heard that many of the people were dying out of heart attack, and then this kind of. So most of the people who were released after the COVID treatment, most of them had suffered from these kind of blood clotting things. So they were put on heparin therapy or any other kind of anticoagulation therapy, let us say, to be precise, and you have to keep it for some time till this all this problem goes. But the point is that, okay, it's good, heparin is used for therapy, heparin is used for anticoagulant to prevent thrombosis during surgery, but then the overdose. So there is a certain dose beyond which, if it goes, then you can have hemorrhage. That also might have heard that, hemorrhage, somebody has a brain hemorrhage, we say, right? So there are certain life-threatening conditions which can come up if heparin goes beyond a certain limit in the body. And that is why it is very important to monitor and control the heparin level. So you need to periodically monitor the blood of that particular patient which is undergoing surgery, that whether the heparin is within that particular level or not. If it is going beyond, then you'll have to neutralize the excess heparin in the body, or if it is less, then you have to add it. So like that. So in this slide, I just tried to give you an idea that why heparin monitoring is required, and what are basically being done with this. So there are conventional ways to monitor heparin, like activated clotting time assay, ion exchange chromatography. Then there are partial thromboplastin. There are various other like clotting assays are there, but they have certain disadvantage. For example, they are indirect, they're not sufficiently reliable because they are indirect and they have interference from some other factors. The sensitivity level is low, they're complicated, time consuming and expensive. So it is desirable that we develop new methods which are sensitive, accurate, simple and inexpensive. And to my knowledge, I believe that fluorescence is the most sensitive spectroscopy, for the very simple reason that if you talk about single molecular spectroscopy, it is so sensitive that you can even detect a single molecule, a photon coming from one molecule. So it is that sensitive, because most of our spectroscopy that we do is like we're dealing with the, millions of molecules. So what we get is an average information. But fluorescence is sensitive to an extent that you can detect a single molecule, right? So that is that sensitive. So if you talk about accuracy and sensitivity, then colorimetry and fluorimetry based assays are considered something of a very which is desirable, I would say. And here you will see that they are mostly optical. So the idea is that these are actually very sensitive form of spectroscopy, and that is why in many of the routine clinical processes, these absorption and emission based methods are being employed. So, well, I am not the first one to target this particular analyte using fluorimetry. There are many who have been doing, and this field has been there for quite some time. In this slide, I'm just trying to say that what are the limitations of existing fluorimetry based methods. So first of all, a large majority of these reported methods, they operate through fluorescence quenching, turn off. I told in the beginning, a turn off is like your emission from the molecule is coming down, is quenched. and turn on is the other way. When you add your analyte, it increases, right? Now, what is the problem with turn off? Turn off has a disadvantage that it is susceptible to large environmental effect, low sensitivity, and false positive result. What do you mean by like low sensitivity and false positive? Just to give you an idea, which I try to often discuss with my colleagues or researchers or students, just to try to give them an idea that why turn off is less sensitive or less desirable. Suppose in a room you have 10, like halogen lamps, big halogen lamps, and then you have a very small bulb was glowing in the same room, which is 11th one, let's say. If you switch off that small bulb, you will actually not see any difference because you have a background of 10 big halogen lamps. But if I put the story in the other way around, you enter the room which is completely dark, and then you put on the bulb, you see a lot of light. Is the difference of a bulb only, is the difference of a small bulb only, but in the first case you had a background of 10 big halogen lamps, huge amount of light was there, so the change is very, very less. On the other hand, you have a completely dark background and then the bulb, so the background makes a bigger difference, and that is why turn on is always desirable because it is more sensitive. A small change you can see, but in turn off, seeing a change is little difficult. I would not say that it is impossible. I mean, there are many great work which have been devised on turn off. It's just about the change that you get, but ideally given a choice, one should go for turn on methods. Now, what is false positive? See, if you take a sample in solution and try to measure its emission intensity. Now you measure after 5 minutes, you measure after 10 minutes, and you'll see whether the signal is changing or not, or reproducible or not. You have not done anything to it, not done anything to it. So suppose, I don't know if some of you definitely have experience with these measurements, you will often see that many a times there is a reduction in the signal, some reduction, let's say 5%, 10%, 2%. Many things can happen. Like for example, say the room temperature has increased slightly, or some of the molecules in the solution has basically deposited on the surface of the cuvette, or wherever you are taking the solution, and or sometimes there may be a fluctuation in the light intensity. So these kind of things will always give you a small reduction in the signal. Now I am telling that no, this decrease in the signal of my molecule is because of interaction with my analyte. But then there are many other factors which impact this and which decreases the signal. So then I might have a false positive. I might have a decrease just because some of the molecules in the solution has basically stick to the cuvette, right? So in that case, I have a less number of molecules in the solution, so my count have the intensity has decreased. So these are basically, so what I'm trying to say that reduction in a signal can happen for many reasons, but turn on is very difficult to get. You often do not get a turn on system. If you do something as casual as this, like okay, you put a molecule in the solution which has not dissolved, and then with time it is dissolving and you are seeing an increase, that's a complete artifact. But I believe that when you are doing research, you are always taking care of those parameters. So this is one disadvantage with the turn off methods in general, I would say. And then the reported methods so far involve complicated and time consuming multi-step synthesis, and which are commercially not available. Severely impedes their prospect in real applications. So what I try to say is that many of the molecules, first of all, let me tell that chemistry is all about making new molecules, synthesizing new molecules, giving them variety of features which somebody desires. That's absolutely nothing wrong with that. And there are dedicated groups, and I think the organic synthesis, if it goes out of the chemistry domain or synthesis, then chemistry will have a trouble to exist. But then when I say that I want to develop a sensor, now the molecule that I want to use is something which has been, somebody has worked for 4 years of his or her PhD and came up with some 5 milligrams of that compound with, let's say, going with 20 multiple steps. Now that particular molecule is amazing in showing response to heparin, let us say. I'm just talking about a hypothetical case. Now for the company to take up this, he will say, okay, how easily is the material available? Then say, no, I think it needs like 3 years or 4 years. They will completely pack out immediately because they don't need it. they want something which we can easily get and which the raw materials are easily available, that needs lesser effort. So even simple things can basically, I would say, turn off your the company person to take your technology ahead. So what we believe, and this idea has been there for some time in literature, that if for a real application, if you can actually manage with molecules which are already established, which are already commercially available, then of course, you have to sometimes you have to compromise with, let us say, sensitivity or selectivity to some reason, right? But then if you can actually found out a molecule which is commercially available with a reasonable accuracy and reasonable selectivity and sensitivity, then it can severely impact its prospect in real application. So what we believe that, okay, if you can have a molecule from the the established base in the literature and which is commercially available, that will really be an advantage. So overall, I just wanted to tell from this slide that there is a need to develop turn-on sensors for heparin, and which preferably should utilize commercial available molecule. So these are the two information that I wanted to give from this slide. So let us just share the story that how we ended up using this particular molecule, thioflavin T, for sensing heparin. So thioflavin T, I've used the word molecular rotor because I'm not really sure the audience who is listening to it how well versed they are with this kind of terminologies. But I am actually trying to address this talk to reach maybe some research scholars also, although it's a faculty development program and most of you have already a lot of expertise in this particular area maybe. Okay, so what is a molecular rotor? So a molecular rotor is a molecule, or it's a class of molecule where you have two fragments and which are basically joined by a single bond. For example, thioflavin T has this benzothiazole group and dimethyl aniline group, and these two groups can rotate around a single bond. Okay, so that's called bond twisting around central CC bond. And if that happens, then in this class of molecules, what happens is that it is a non-radiative process. So I'll just briefly talk about what is non-radiative. So ideally what happens, coming back to the Jablonski diagram, so you excited a molecule which is in the ground electronic state, it went to the excited state here. Now whether the molecule will directly come back through fluorescence, or it may also give like non-radiative processes. So when you excite a molecule, whether that molecule will emit or not will depend on what is happening to the molecule in the excited state in the form of non-radiative and radiative process. If the radiative process will predominate, you will see a emission. If the non-radiative process will predominate, then you will not see a emission, because that energy goes in the form of heat to the surrounding. Okay, so if you want to get emission from a molecule, then you have to cut short its non-radiative processes. So for this particular class of molecule, this kind of bond rotation around the central CC bond has been established to be a non-radiative process. So if you can block this rotation, somehow you can actually stop that process, then you will actually cut down this non-radiative process and you will force the molecule to emit through fluorescence. So ideally, a molecular rotor class of molecules, the emission intensity from this molecule is heavily sensitive. So I spoke about, so what we found out is that this molecule has a 1 picosecond lifetime. That's very short, to be honest, because if you remember my slide on lifetime measurements, I used a word called time correlated single photon counting, TCSPC. But the resolution is you cannot measure anything below let's say 0.2 nanosecond. 0.2 nanosecond is, let's say, 200 picosecond. So anything below that you cannot measure. So if your molecule has a lifetime which is less than 200 picosecond, you simply cannot see. And I mean, most of the university laboratories even do not have access to like, let's say, TCSPC. So forget about ultra fast thing. But being at BRC, that advantage we have sometimes. And so we this work I did in my basically PhD, and we found out that this molecule decays very fast. It's 1 picosecond. That's the time scale. And if you look about the quantum yield, then quantum yield is again 0.0001. What does it mean? That if the molecule absorbs 10,000 photons, then out of those 10,000 photons, only one will emit. And this is the condition in the bulk water, because in the bulk water, the molecule is twisting very fast. So when I say that the molecule is twisting, what is the conventional way you can immediately think of stopping this rotation? I would say that, okay, let's put a higher viscous medium. Let's put that molecule to an higher viscous condition. When you put the same molecule in the higher viscous condition, what will happen? This bond rotation, which was happening very fast, will now slow down. Now, when it slows down, then that means what? That these non-radiative processes are being cut short to some extent. So the molecule is bound to stay there for some more time, and then you will have more and more fluorescence. So if you look at the literature, then basically this viscosity test, we can say viscosity dependent measurements. So that will tell you that whether the molecule has a molecular rotor nature or not. Because sometimes by looking at the molecule, you feel that, okay, I think this molecule is a class of molecular rotor. Why? Because, okay, it has single bonds and the bond rotations and other things are possible. But then you'll have to show through viscosity measurements, viscosity dependent measurements, that whether this molecule belongs to the class of molecular rotor or not. So for this we have established, so I have written viscosity controls the emission yield of thioflavin T. So as you can see that as we increase the viscosity, the emission yield or emission intensity increases. Okay. So what is happening? We are basically stopping the bond rotation, or we are actually getting slower and slower. See, this is decaying much slower compared to the red lines, like that. So this tells that this molecule this is strongly dependent on the viscosity of the medium, and we can say that it is a molecular rotor class of molecule. These terminologies I have been using this in this entire lecture is common terminology like amyloid fibril. Time and again I'm coming to it. So as I said, like amyloid fibril is basically sort of a a protein aggregate, and so this is a typical structure of these amyloid fibrils. Now thioflavin T is known to be a sensor for amyloid fibril. It's a gold standard. I don't know, maybe some 40 years back this was discovered that this molecule gives strong emission response in the form of in amyloid fibrils. And since then, any new molecule you develop for amyloid fibril detection, you will have to show it, compare it with thioflavin T, because that's considered as the gold standard in some sense. But if it would have been like a real gold standard, why would people develop new molecules? So the idea is that, okay, it is so well established that you will have to compare your result with thioflavin T and tell that, okay, this is how my system is performing with respect to that. So these are particularly, but my discussion is that, okay, thioflavin T, when you put it into the fibril, it gives large emission enhancement, established 50 years back. No difference in that. It gives very large emission, almost of the order of 1,000-fold. Okay. Now, what is so special about it? Nothing is special. But then people have been debating that in what form thioflavin T binds to these fibrils. Is it monomer binding, or is it like the aggregates of thioflavin T is binding to the fibrillar system? And while this discussion was there in the literature, so there's a lot of debate. So some people felt that no, the monomer only one particular form of dye is going and binding. Some people said no, it is the aggregate binding. So while this debate was there in the literature, we tried to establish this, or I would rather say solve this controversy in a different way. What we do, we took a polystyrene sulfonate. Polystyrene sulfonate, you would know, it's a polymer with a lot of negative charges around it. So it's a polyelectrolyte nature. And then we tried putting thioflavin T. But why we choose polystyrene sulfonate? So polyelectrolytes are known for inducing aggregation. How it induces aggregation? Now, normally, if you take a cationic dye and if you put in a free solution and you put another cationic dye into it, do you think that they will bind together and form a dimer or aggregate? An aggregate is what? It's a collection of molecules, more molecules coming close together, right? Together. So mostly these aggregates we see in micelles, surfactants, hydrophobic associations and things like that. But if I'm talking about two small cationic organic dye molecules, can you believe that you can actually make them aggregate? The answer is yes and no. Okay, let's say why it is no, because I would say that this is, there is an electrostatic repulsion. Two cationic molecules cannot club together. Fair enough. But then if you say that, okay, I have a way to do that, I will increase the concentration. I will keep on increasing the concentration till it's soluble. So then at some point of time they are bound to aggregate, because you are not leaving any space for the molecules to stay. So aggregates you can find specifically when you go to very high concentration of dyes. So that's a separate analogy. But in the case of polystyrene sulfonate, what happens is that when one dye molecule binds to this negatively charged surface, then there is a charge neutralization, because then this cationic molecule cannot be treated as if it is barely cationic only. The charge density on it reduces, right? Okay. Now, once that happens, then I can have another thioflavin T coming and binding here, because the main problem was with their cationic charge. Now, once that is neutralized, they can stay together, right? So basically, these are the media which are known to induce aggregation charged dye, and that is precisely we try to do here. So when we put thioflavin T in polystyrene sulfonate solution, we see that there is a different band. And if you can, I would like to draw your attention to the peak position. So PSS, its peak position at 550 nanometer. Whereas if you look at the amyloid fibrils, this is around 490 nanometer. So see, there is a distinction between 490 and 550. So, and we tested with several other systems where actually aggregation would have supposedly taken place, and then we realized that in any other in all these systems where aggregation was supposedly forming, we see a distinct emission band at 550 nanometer. So that tells us very clearly that in the fibril the band is not at 550, it is at 490 nanometer, and that is why this molecule has to bind to the monomer form in the fibrillar matrix. So we tried to solve this question, that whether it is a monomer binding or an aggregate binding, by just seeing the peak position of the emission spectrum, right? So in a nutshell, what we conclude that polystyrene sulfonate induces aggregation of TST molecules, and this controversy of which way the molecule binds to the amyloid fibril is kind of sorted out. But then what was the message for us? The message was, okay, so that means aggregates of TST can be formed, and they are basically associated with emission band at 550 nanometer, right? So with this information, we looked into the literature. Am I audible, right? Because sometimes the internet goes, I don't have a mobile here to connect.
So you are audible.
Great. Okay. So yeah, so if you look at the heparin structure, so I gave you a complete analogy of why we selected polystyrene sulfonate. So when we looked at the heparin structure, we saw that, okay, this is basically told as the most negatively charged biomacromolecule known in the literature, more negatively charged than DNA also, because DNAs are also considered negatively charged because of the like negatively charged phosphate backbone. So then when we looked at the structure, then we thought, okay, why not we put thioflavin T, and looking at the structure, it seems that it can give us an emission band at 550 nanometer. So this is what we did. We took a thioflavin T solution and then we added heparin into it in a gradual manner, and you can see that there's a distinct band at around 550, 560 nanometer. So the free form of the dye is, I would not say non-emissive, but a very weakly emissive species in the free monomer form, whereas in the presence of heparin, we have an emissive aggregate at 560 nanometer. And we had an emission enhancement of around 45-fold. So that's a huge emission enhancement. Then we looked at the absorption spectra. In the absorption spectra, we also see that there is a gradual shift and there is a decrease in the absorbance. As I was telling in the beginning, that when you are looking at the interaction between a molecule, your probe molecule, and the analyte of interest that you want to measure, then you see certain features, and those observables are in the form of emission intensity, emission peak position, absorbance peak position, absorbance intensity or optical density, whatever you say. So in the case of TST and heparin, we see that there is a blue shift in the absorption spectra, and these and then basically boils down to somewhere at around 393 nanometer. So again, like maybe that is a more involved and dedicated discussion, but maybe for the timing we can go ahead with some of the known, I would say terminology. So when you are dealing with aggregate, either the molecule has a blue shift in the absorption or red shift in the absorption. Now, in general, the blue shift are associated with the H aggregate formation, and the red shifts are normally called J aggregate. What is an H aggregate? An H aggregate is called a head-to-head arrangement of molecules. So molecule can aggregate in multiple ways. It can aggregate in multiple ways. But when you have a blue shift, then we can ideally say that, or not say ideally, but in general, that this is a sort of an indication of H aggregate formation. So that is precisely what this feature seems like telling us, that you have a H aggregate formation of thioflavin T molecules in the presence of heparin. Then I went for measurement of excited state lifetime. So as you can see, the dye in the free form is in the very fastly decaying, whereas when you put it in the heparin, so that means when it is in the aggregate form, it is very slow. So you see the emission goes up to somewhere around 9 to 10 nanoseconds, whereas monomer it is in within the pulse, very slow. So that tells us that the emission average lifetime from somewhere around 1 picosecond. So in the aggregate, it got around 1.4 nanosecond, very slow. So there's a very large modulation in its excited state lifetime. Then going to the notion of this is not something that is related to absorption and emission, but again, just to complete and complement the story that we are trying to make here, is that we did CD spectra. Many of you must have done CD measurements. So what happens is that you have what we see is a clear bisignate feature here. So this kind of bisignate feature in circular dichroism spectra is basically an indication that you are having excitonic coupling, which is nothing but aggregate. So what is excitonic coupling? So you have aggregates. So when you excite the molecules, since the dipoles are very close and interacting, there is an excitonic interaction between them, and that leads to this kind of splitting. We'll not get into detail here, but just to give you another evidence that THT forms aggregate in the presence of heparin. Then there are many other ways, as I said, and it is important for the discussion here also, that temperature dependent measurements we did, and what I'm showing is here your emission spectra and lifetime. So in the present story, how temperature dependence fits. So what we proposed, that we have an aggregate of what? Thioflavin T in the presence of heparin. So it's basically a non-covalent assembly. Thioflavin is basically associated with some electrostatic interaction, and then THT molecules will have some pi-pi interaction among them. Now, these kind of non-covalent assembly are known to be sensitive to temperature, because these are weaker interactions. So you cannot break a covalent bond with temperature. Probably you can, but you really need to go to very high temperature. So non-covalent assemblies are known to be sensitive to temperature. So we tried testing our THT heparin complex with the temperature. So you see what is happening when you increase the temperature, the emission intensity goes on decreasing. Similarly, excited state lifetime, if you see, then that also goes on decreasing. What does it tell? So it tells that when you increase the temperature, it is possible that the THT aggregates breaks away from the surface of heparin, because you are giving more energy to it, and they were kind of weakly held. So they try to come out of the system. Once the thioflavin T is free in the solution, then emission intensity will be less. So with increase in temperature, we are kind of breaking those aggregates, and that is reflected in the form of emission intensity decrease. Similarly, in the lifetime, as we saw that when there was an aggregate, there was a very long lifetime. When we increase the temperature, we are kind of breaking those systems. So once it is breaking, the THT comes into the free form, solution form, and then it does not have a very large intensity or very short lifetime, right? So then you see that there is a decrease in lifetime. Now, I was also again telling in the beginning that when you try to develop a sensor for a particular analyte, then you make sure, or at least you ensure, that your probe, your reporter, your molecular probe should not give similar changes with some other analyte. And that is what precisely this slide, that's called selectivity. So that is true for most of the sensor work. You have to show selectivity. In this graph, basically what I have been trying to show that there are structurally related analytes with respect to heparin. That's called chondroitin sulfate and hyaluronic acid. So these are all glycosaminoglycans, but there is a charge density difference. If you look at then this particular heparin, this is a dimer repeat unit because heparin is a polymer, right? So this is a dimer repeat unit, and then you have 4 negative charges, 3 in the form of sulfonate groups and 1 in the form of carboxylate group. When you move to chondroitin sulfate, then you see 1 sulfonate group and 1 carboxylic group. So the charge density per dimer repeat unit is reduced to -2. So these are -4 negative, this is -2, and hyaluronic acid has all neutral groups except this carboxylate group. So if you look at the charge density, -4, -2, -1. Now, when we put the same concentration of these 3 analytes with the thioflavin T solution, we see that there is only a decrease in the case of increase in the case of heparin. Others did not really respond well. So suggesting that the charge density in the heparin plays a very important role in making the interaction very selective towards thioflavin T, and that too, as I said, that when the charge density, negative charge density is high, it probably induces aggregation in a very sensitive and selective way. Then we also tested various other analytes because that is part of the protocol. You'll have to show many other analytes, and we saw that the maximum response that we could achieve was for the heparin. And then these are the things you'll have to evaluate and tell when you're talking about sensor. Then you'll have to tell that what is the limit of detection of your method. That means what minimum concentration of the analyte that you can detect. So we kind of do this kind of calibration plot, and then from there we can find LOD. LOD mostly, I don't know, I should define here 3 sigma by S we normally say, that I have not written here, but 3 sigma is what is standard deviation for 10 blank measurements. So there is no heparin, and that solution you measure 10 times and see what is the standard deviation of the measurement, and then you have a slope here. So from the slope 3 sigma by S you can find these kind of figures. So again, that is in the aqueous media, right? So the idea is that okay, we have to be able to measure. We have done nice spectroscopic features we saw, and then we found that okay, there is a very nice 550 band is coming on, and only showing response with respect to heparin. And life is really simple, cool, and accomplished. And what else you can expect? And we actually published this paper in Applied Materials and Interface. But the life is more complicated when you go to the biological media, because heparin is not something that you would like to detect in water. You have to inject it in the blood, in the serum, and then that concentration has to be found out in the serum matrix. So the matrix is very different, and that is precisely where most of the sensors struggle a lot. Everything is good in water and buffer. Real matrix, real problem, real challenges, and that is where all of the people working in the sensor area is right now, I would say, is struggling. I mean, this struggle has been long existing, but still. So you have to really find out something which can actually work in the real serum matrix. So for that reason, you have to evaluate your sensor in the complicated biological media, and you have to do it in the serum matrix. And these 2 things basically has driven my research from 2016 to 2022. I mean, yesterday only I got a paper accepted in sensor and actuators on heparin, and this was my first paper in 2016. So 5-6 years I have been doing this research, and just by designed by this particular media, that at what percentage of human serum matrix I am able to identify heparin. So I started with 1%, I would say. So this particular work we have only could detect in 1% serum matrix, and the paper that got accepted yesterday, there we could achieve in 100% serum matrix. So as I said, so this has been a real journey. Now there are some other aspects to this work. So this is a very a talk which basically tells about or needs to discuss about various aspects of absorption and emission spectroscopy, and what kind of information that we can actually get out of these 2 spectroscopy. So this title I have put as heparin protamine interaction. Why? What is the need to study heparin protamine interaction? So in one of my very previous slide, I said that heparin is all good. It is used for anticoagulant. It is used for deep vein thrombosis. A lot of good therapy being done with heparin. But then if it crosses beyond a certain limit, then it is dangerous. It is life-threatening. So you have to monitor, and then suppose if you find that there is a heparin overdose, what are you going to do with it? So you have to neutralize it. So protamine is a arginine-rich polycationic peptide, which is the only approved drug for treating heparin overdose. So that is where the interest in protamine heparin interaction comes. So heparin on the one side is a very negatively charged macromolecule, biomacromolecule, and protamine on the other end is a cationic peptide. So naturally there is a electrostatic charge neutralization, and once the charge of heparin is neutralized, then it will not be able to do its regular activities in the form of anticoagulant. So protamine is normally administered to treat the heparin overdose, and it basically reverses the effect of heparin. So we thought that whether can we monitor heparin protamine interaction with our probe, because you have, okay, so I think the idea is here. So you have this heparin and diaggregate system which is emitting. You put protamine into it, you expect protamine to bind to heparin because protamine is much more cation rich. It's a very highly cationic peptide, and in that process it will bind competitively and knock out the dye from the system. Once the dye is knocked out of the system, then it will, the glow has gone. That means what? The emission is decreased. So this has been shown here in this particular form. So once we had the complex, then when we started adding protamine, then the intensity decreases. So it basically signifies this kind of situation happening. In the case of lifetime also, we had a THT heparin complex, and then we add protamine, and it goes back to this kind of form. And then in the absorption spectra, we see a sort of recovery, although there was a background, because once heparin protamine binds, then basically you have a big macromolecular solution system in the solution, and they often try to precipitate it out of the system, and that is why this kind of turbidity you will see in the absorption spectra. So the background becomes high. Now another direction that we try to explore from this thioflavin T heparin complex is detection of heparinase. Heparinase is an enzyme which specifically degrades heparin, and it is used to produce low molecular weight heparin for therapeutic purposes. What is therapy? So basically, as I said, like heparin is a polymer, and sometimes it has been seen that clinically low molecular weight heparin, something in the range of, let's say, 4,000, is considered more important. So you would like to cleave them. That is one of the purpose. But other than that, heparinase enzyme is also a biomarker for several diseases such as rheumatoid arthritis and diabetes. So there is a need to devise methods for detecting heparinase. And when we, at that point of time, when we looked at the literature, then we found that there are limited reports for sensitive and selective assay for heparinase. So the idea was something like this. Again, since this is a diheparin complex and heparin is an enzyme, so it is going to cleave it. It is going to cleave its backbone like anything. Once these are put into smaller fragments, then the charge density is not there. When the charge density is not decrease, as I said in the beginning, that it's not always that the turn off is not done, because here the diheparin complex is emissive, so the heparinase has to be cleaving them, and if you want to develop an assay for heparinase, then it is basically a turn off detection. Then we also did some sort of selectivity assay. Then we have to test several enzymes, and then we saw that heparinase shows the maximum response for this particular system, thioflavin T heparin system. So in conclusion, for this particular part, we have to develop the quick and robust turn-on system for heparin detection. Of course, it was having an H aggregate, but I'm not going on the implication of why H aggregate and whether that makes any importance or not. But one line I would certainly like to say, that H aggregates are generally considered as non-emissive, and that is the point I tried to focus in that research article when I wrote that H aggregates are considered to be non-emissive in nature, because once you have head-to-head orientation, in case of, let's say, like planar aromatic molecules, then they have several pi-pi stacking interaction. They are again considered non-radiative. So if that happens, then normally these are non-emissive. But in the case of molecular rotors, if it is H aggregate, so because of this aggregation, what happens is that rotation, which was previously happening very freely, is frozen. And if that is frozen, then you have emission response. So there are two things that is happening. Although it is an H aggregate, but since this molecule was able to freely rotate, and in that case, when it has a lower emission intensity, so that part has been blocked. So because of that, you have more emission coming out of it. But ideally, if it is not a molecular rotor and you form a H aggregate, then you are going to see quenching of the system. So this was a very rare example of emission coming out of H aggregates. Anyway, so like it was selective, and then we could actually also apply to the complex biological media, but make sure that it was just 1% serum matrix. And as I told in the beginning, then 16 to 22, 1% to 100%. That's the journey. So this result we could actually do it with only 1%, and we could actually measure both by absorption and emission. We say colorimetry and fluorimetry. And so yeah, so this was the story for thioflavin T heparin. And as I said, like we also used this particular platform for measuring heparin protamine interaction, and then like for heparinase assay. Now yeah, just a minute. Yeah, so there are a few other probe molecules. So what happened when we actually were struggling with the serum matrix? So serum matrix, we thought, okay, first of all, we thought maybe this particular dye has a issue in responding to the serum. Maybe if we take some other chromophore, okay, and with the same features. This is thiazole probe. It's again monocationic. I have put a charge here just to show you that this is cationic. And then we thought maybe thioflavin T has some issues. So maybe if I take a similar class of molecule like cationic only, but some other chromophore, then maybe it can perform better. So with that thing in mind, we tried exploring various cationic molecules. See, we will be very, very clear. So I'll put you two directions in the mind, and then maybe you will follow up this coming up discussion in a better way. One thing that has to be kept in mind, that we are looking for fluorophores which has to be cationic, because heparin is negatively charged. And if heparin is negatively charged, so the dye molecule has to be cationic, because then only it will be attracted and it will bind. Okay. And the whole idea is that we are looking at the aggregation of those chromophores, because there is an idea of aggregation induced. These are all molecules which are some sort of molecular rotor. These are all molecules which are, when they're free, they're able to freely rotate. But when they're aggregate, then they cannot rotate, and then you have emission response from the system. Why? Again, because these concepts I repeat again and again so that you follow it up better. That if the molecule freely rotates, then that's a non-radiative process, and if it is a non-radiative process, then it will de-excite, or it will basically allow the relaxation of the excited state to come down to the ground state in the fastest possible way. So if you aggregate them, then now those processes are blocked, and then you will have emission from the system. So these two directions we kept in mind, that the charge on the probe has to be cationic, and then we tried exploring different probe molecules just to see that whether if it responds better or not. So this was another probe that we took, thiazole orange, and if you see that there is an emission response, the emission intensity goes on increasing with increase in heparin, and we have seen in the case of thioflavin T also. So I'm not explaining in detail here, but just to tell you the basic preamble is that, see, the free thiazole orange has a band at 530 nanometer, whereas it binds to heparin, then you have a 630 nanometers. So see, there is a rate shift of 100 nanometers. So beginning I was telling, measuring the interaction of probe with some analyte of interest, then you will see the changes in the feature, the peak position, and then you will see the changes in the emission intensity, and those spectroscopic observables helps you to define the problem in a better way. And so again, we see a emission enhancement of 22-fold. And this line, I think by now you should be able to follow that heparin induces aggregation of thiazole orange due to charge neutralization of cationic thiazole orange. Idea is, as I said in the beginning, that these dye molecules will not aggregate by itself, but when you put it in the presence of heparin, then these charges are neutralized, and then more molecules can come close together. Then absorption measurements we did for the same system. See, nice absorption features compared to the previous dye. So this is the monomer form which basically goes down, and this is the aggregate form which comes up. Okay, so this is so when you add heparin, so this band goes down and a new band comes up, and this we are allocating it to the aggregation. And if you plot the features with respect to the heparin, consider nice linear plots with this one. So these you can utilize for sensing and drawing linear limit of detection and things like that. So similar to thioflavin T, now we have a higher blue shift, and again, as I said, blue shift in the absorption maxima is a characteristic of H aggregation. And if you look at the color change also, so that basically tells that you can possibly have a naked eye detection here also. The naked eye detection has its own advantages. See, when there is a word called resource constrained conditions. See, you want to develop a method which has to operate in a village, and they do not have a lot of equipment. Let us say a very small clinic and things like that. So if you have a method basically which can actually, I'm not talking with respect to the heparin, because in a village you will not do a surgery, right? I mean bigger surgeries. But in general, I'm just trying to tell you that why naked eye detection is important. So in a resource constraint condition in a village, a very small setup, so something which you can actually develop a protocol that by looking by naked eye you can tell, okay, this is positive, this is negative, and things like that. So that really helps, right? So there is an advantage of naked eye detection in many other ways. So in this particular case, we could actually make that advantage. And I will not go to CD measurements, as I said, like it has a bisignet feature. But again, in the lifetime, you see that there is a large variation in the lifetime. So these data are sort of repetitive, but it also tells you that the same kind of features are happening. So the phenomena that you are seeing is similar. Now you will have to evaluate the performance. Now we have understood how it interacts. Now we have all nice features in the absorption and emission spectra. Now, but we have to tell that, okay, whether it is sensitive, whether it is selective, whether it goes beyond what we have done previously. So that is where now the things have to compete. And then we have done effect of ionic strength on thioheparin complex. I think this particular part I did not tell in the beginning. So just to tell you a little bit on this. So what will this experiment tell us? Effect of ionic strength on TOF. What will this experiment tell us? I have a complex of the dye with the heparin, and then I am putting salt to it. What information it will give us? So the idea is that we are assuming, and from the beginning, I think I made your mind biased towards it, that I have a negatively charged surface on heparin, and I have a cationic dye, and both are going and interacting, and there is a electrostatic charge neutralization, and then more dye molecules are coming, and in the process I have an aggregate, and those aggregates have its own spectroscopic features which is distinct from the free dye, and these features I am trying to utilize for sensing heparin, right? Now there is an assumption. What is the assumption? The assumption is that this is cationic and this is anionic, and both will go and bind through electrostatic interaction. This is a very obvious assumption. But suppose I ask you that, is there a proof for it? Well, so there are many other ways to do that. One obvious and reason, as being a researcher in the field, you would say that I would do zeta potential measurement. What would the zeta potential basically measures the the potential, the electric potential on the surface. But if I want to tell you that, okay, I have to measure through absorption and emission only, I don't have access to zeta potential. So what will you do? So the idea is that since it is an electrostatic interaction between a cationic and anionic species, so if you put salt into it, ionic strength, if you increase, it will screen the interaction between the interacting partners. So if it screens the interaction, so what will happen? Then it becomes weaker. Once this interaction is weaker, then the complex will break. And that is precisely what we see in this case, that when you increase the salt, we see a decrease in the emission of what? TU and heparin complex, thiazole orange and heparin complex, and this goes on decreasing. Okay, so that means what? That the salt kind of screens this interaction between the two, and which is presumably nothing but electrostatic interaction. So this is in one way we are trying to establish our assumption, a fundamental assumption that we made in the beginning. But later on, this experiment has far-reaching implications. If you talk about the serum matrix has a very high ionic strength. Now, that also tells us that why thioflavin T heparin complex did not work in low concentration of serum. What was the reason? The reason was ionic strength. Because if you have electrostatically assembled system, the probe and the heparin is electrostatically assembled. Now you are putting it into a medium which has a very high salt concentration, then this interaction is getting weakened. So in a real matrix it will not work, or at least if it works, then at least there will be limitation in working. So ionic strength test is also important for us to understand that how strong my system can survive the ionic strength of the medium. So we'll come to it later on, and we will show you that how actually we could get rid of this particular thing. Then effect of temperature, I think I told in the beginning, so I'll just skip this slide so that I can tell you newer things. But this was just to tell you that this complex breaks when you increase the temperature, and which has its feature in the absorption and emission. Because the title of the talk is absorption and fluorescence spectroscopy, and I'm trying to tell you that there are multiple ways and multiple tests that you do to your solution, and all of them are reflected in the most simplest form of spectroscopy, which is absorption and emission spectroscopy. And the observables that we are always looking at, the peak absorbance and the emission intensity, emission peak position, and things like that. Now we went on to evaluate this sensor system in serum samples. So my parameter after that publication 2016 was that can I have a system which can survive more in the serum samples. So you look 1% to 5%, that's all we could achieve in maybe some 2 years. That I have a new probe which works in 5% human serum. Then selectivity assay we did, and in selectivity we could see maximum response for heparin. Of course, others also responded, and that is where, as I said, like when you bring a molecule for sensing a particular analyte, you will have to look into many perspectives. Like it worked in serum, but it had, let us say, somewhat compromised selectivity, I would say. Then another is pH range of operation. So the probe that you are using should be able to operate in a large pH broad range of operation. Protamine sensing I already said in the previous work. So something similar here. So you have a thioheparin complex, and when you add protamine to it, it breaks. Similarly, you see very nice absorption changes. Initially this band was decreasing, now it is recovering because the dye is broken from the heparin complex, so it is a free dye, so it will show a feature of free TO. So these kind of reversal in trends happens when you add protamine to a thioheparin complex, and this basically tells that you have a disassembly or breakage of TO aggregates from the surface of heparin. And then anyway we could publish this work, although we made a very small I would say progress in terms of performance in the serum matrix, but it is difficult, and that is why these kind of small progress is also acknowledged in these journals. Then as I said that H aggregate is something which is non-emissive in general, but since it was a molecular rotor class of molecules, so I had some emission. So then we realized that is it possible that we go to J aggregate, let it be molecular rotor based only, but if we make J aggregate, in that case we can have a higher sensor performance, a better sensor performance. And indeed it was the case, and in this particular case actually we took up a molecule which is a textbook material, I would say, pseudoisocyanine. This is a dye known for ages, and there is a word called repurposing of drug. I mean, you most have heard during these pandemic times, the same drug is being used for different purposes, right? So it's sort of a repurposing. This has been established for a different reason or a purpose, but then my intuition was that okay, this structure seems to be happening there. So let's say if it is a J aggregate, normally it forms J aggregate. So let's see what happens in the case of heparin. and J aggregate normally gives higher emission, so maybe we can achieve something better. So indeed, actually, we could emission enhancement. Just so this band decreases and this band goes up. And I told you in the beginning that when there is a blue shift, that's H aggregate. When there is a red shift, that's J aggregate. So here we have a from the monomer to the red side, there is a distinct J aggregate band. Then we characterize in a routine way, because if something new is you are trying to propose, you have to tell and so every single thing in terms of its characterization. So we do temperature dependent and then circular dichroism, and of course the protamine thing. So by now I think then in selectivity analysis also we could really make something good data out here. So you see the heparin also again shows a very high and selective response with respect to its related analytes. And in the serum matrix, well, we went up to 5% again. So we were limited to sort of 5%, and although we could go to J aggregate, so then we realized that it is not possibly this nature of aggregate H and J. And one indication that we got out from previous data was the ionic strength. We realized that ionic strength tells you about the electrostatic interaction between the di and heparin. Now my heparin is fixed. Heparin I cannot do, it's a biomolecule that I'm trying to understand. What I can play with is my probe molecule. Now and so far whatever molecules that I dealt was all monocation. So then we thought, if we increase the charge on the molecule, the probe molecule, the reporter molecule, the fluorophore, and if that can help us in improving the performance. So we made a, like, I will not say quantum jump, but a small jump from here. But we did not wanted to lose on other, parameters. Yeah, just to conclude this J aggregate part. So again, like, see very nice features, and again, getting into a very good journal like Sensor and Actuators. So it has, again, as I said, like these fields are competitive. Now the challenge was limited performance serum matrix, and as I made this story, so now we thought, let us take a dicatonic probe and see whether we can improve the performance. And these are, again, I am not going to change the molecular rotor nature of this. And what I have done is, like, I have just increased the charge, and these are again commercially available. So I have tried to keep those parameters in my mind while I choose a molecule. And with this, I went ahead doing more measurements, and then you see a fluorescence increase, and then the selectivity, rate shift. So I'll just keep on telling that, look at the shift in all the cases. So that tells that interaction is happening. But now my judgment to the result is performance in the serum matrix more or less, while not compromising on other parameters very heavily. So in absorption measurements, again, very nice changes in the absorption spectra. And again, in the said, like, you tell something new, you have to establish everything that you do. Although it might seem very easy, obvious, but then it has taken a lot of hard work and effort to reach those measurements. So now in the serum matrix, when we went, so there we actually could find something in the 10% human serum. So see, we have increased the charge from monocationic to dicationic, and now we see some improvement in the performance in the serum matrix. So we could now work to 10% of human serum. So we were kind of happy with the result, and then we thought, okay, let us, now that gives us a way ahead, way forward, and now we can probably do something better. So selectivity wise, if you see that the UPRO1 is quite selective towards heparin, and something that we could actually achieve with this particular molecule. So we tried publishing that. Now we thought that, okay, now we really need to increase it further. So we went to tetracatonic. So we now, since we have got direction, so we thought, let us increase the charge. And this was achieved from one of our collaborators. We are collaborating on a different aspect. Then he had this inventory of molecule, and this particular class, I said, like, maybe I would like to test, although he synthesizes them left and right, but for me it was something different to look at. So then we did all these basic measurements, fluorescence measurements, selectivity assay, Again, the same story of heparin induced aggregation of this particular tetracatonic probe. The only difference is now it is tetracatonic, and of course the chromophore also changes, and very strong fluorescence is achieved. See in the dark, this is just free TP. I mean this probe, and this is in the presence of heparin. Very nice changes in the excited state lifetime. And then, as I said, like we did, in terms of protamine sensing and ionic strength. So I'll probably focus slightly here. So you will see that in previous cases, we could not actually survive something around 10 millimolar of NaCl. Now in this particular case, we could go somewhere around up to 120 millimolar of NaCl. So 120 millimolar of NaCl, so that gives us a confidence that, okay, now I think this particular probe can actually work in high ionic strength media. So there is a possibility that we can actually have a better performance in the serum, because that is the real matrix where it has to work. And as you can see that we could get some response in the 50% human serum with this particular probe. So the conclusion is that now with the utilization of tetracatonic probe, we could actually devise a turn-on sensor for heparin with a LOD of around 6.9 nanomolar, and it shows improved performance in human serum matrix, that is around 50% serum matrix. I forgot to give the bibliography of this work. This is also in Sensor and Actuators in 2022 only. Then we thought, now we did not have a molecule which goes beyond tetracatonic, although we are still collaborating and asking those guys to make, because they can make it. So we went for a tetracatonic porphyrin probe. This is commercially available, and porphyrin are known to be very robust. And then I looked at this structure and then say, okay, this is also having a tetracatonic structure. So let's see. Like, because see, we use the same monocatonic probe, but then in some cases we had 1%, some cases we had 5%. So then we thought, let us see how the interaction goes. And then this particular probe we established through fluorescence and absorption. And those who are working in the porphyrin would probably understand these features better. So in this particular case, we had a turn off response. So that is where we thought that, okay, let us see, even if it is turn off, since it is tetracatonic and the platform is very robust, so maybe that can help us in some way. So we did this measurement with tetracatonic probe, and then lifetime measurements, CD measurements, and then ionic strength. So I will probably focus here on ionic strength. So again, you see this we could actually go up to 210 millimolar. So ideally, this particular probe with heparin could sustain very high ionic strength of the medium. And then we thought, okay, now I think there is a hope that it can work in the full serum. So we went ahead and we tried to evaluate this probe for 100% serum samples. So we could get some reasonable response. I would say 5.2 micromolar and 8.3 micromolar. But then let me just quote you this literature, that the performance of heparin sensing probes in 100% human serum is rarely reported. I think there is only one probe, if it is reported. Let me show here, Mallard blue. Yeah. So there is only one probe so far in the literature that works in 100% serum. That's Mallard blue, and it was published some 7 years back in JACS, I would say. It was a synthesized molecule, and which has a only turn off response and only changes in the absorption. No emission features, nothing. Only absorption changes were there, and they could report in 100% serum samples, and their LODs were somewhere around 11 to 12 micromolar. So here in this case, we had a probe actually we can detect both through absorption and emission, which works in 100%, and at the same time it has a better LOD. Then protamine sensing we have also established, so that I'm not discussing here. So in conclusion, we have employed a porphyrin based tetracatonic probe molecule for the dual detection of heparin with a LOD of around 11 nanomolar. It works in highly competitive media, that is 100% human serum samples. And as I said, like, it is simple, economic, because it is commercially available, dual readout, both absorption and emission, and utilizes a commercially available molecule. So this was the I would say the best work that we could do in this field, particularly, to be honest. And this only got accepted yesterday. And so the idea again, as I would say, that of course it works in 100% serum, but there is still a hope that we can actually reduce the LOD, we can make it even better. And as I said, it is a turn off. So we are looking for molecules where we can achieve the performance in 100% human serum samples, but then in a turn on way, in an increase in emission. So that can probably, possibly help us in achieving a better limit of detection. So with this, I would acknowledge my colleagues at BRC, and these are the two students who have extensively worked, especially Srishti. I think her most of the PhD work is here, and this is my address which is given here. And if you guys feel like discussing further, then I might discuss few more things, but these are all like mostly discrete ways, like one particular analyte. But through this particular discussion, I just wanted to tell you the strength and power of these two spectroscopies. If you choose your molecule in a proper way, if you choose your analyte in a proper way, then you can probably do many things with it. And just to probably finish this talk, since I have put it here, as I was telling in the beginning, that there are variety of analytes that you can detect. Heparin is a one full story that I ended up saying. And then the serum albumin, I was telling in the beginning that in the urine, if you have very small amount of albumin protein comes up, then that's an indicator of early stages of liver and kidney related diseases, something called microalbuminuria. And if you have a very sensitive probe to detect in urine matrix, then this is something that can work. So we developed this particular method. And then I also spoke about perrhenate anion, which is a non-radioactive surrogate of hazardous pertechnetate. And there also we took a probe where actually we had aggregation induced emission. All this work is all separate story, I would say, 3-4 works, and it has its own history, but I just thought that I probably could not finish all of it. So I think maybe I will take some more time for the discussion rather than telling more and more. But I hope that through this discussion, I was able to convince that this is a very interesting and exciting field, and many more things can be done. I think I will stop here, and I would be glad to take questions from the audience if they feel like asking.
Thank you, sir. Thank you for the very informative session. Now the stage is open for the discussion for question answers. Anyone who has any doubts or any questions, sir is here to answer us. Sir, I have one question. In the end, you showed us two molecules. One is both were tetracatonic, right?
Yeah.
One was showing a turn off mechanism and other was showing us turn on mechanism.
Right.
Any kind of discretion you can make on what grounds they are showing different kind of behavior?
Yeah, so if you look at the molecular structure, the one that was showing turn on, maybe I can go to the slide. Just a minute. This one showed turn off response, right? So this is a porphyrin molecule, and this does not belong to the class of molecular rotors. Okay, so these guys are very strongly emissive in its normal free condition. So once the dye is emissive and you do not have a large amplitude, see the molecular rotor. Okay, maybe if I go to the structure here, you will get a better idea. Yeah. So these molecules they belongs to the class of molecular rotor. Basically rotates in the excited state. And if you are aware of tetraphenylethylene, this is a plethora of molecules have been developed on this particular chromophore, which are called aggregation-induced emission chromophores. So these chromophores are known to show large emission upon aggregation. And so these are typical turn-on probes. So anything if I develop on this particular framework, they are going to be turn-on probes only in case if we are able to make them aggregate. Whereas in the case of porphyrin, they do not belong to that particular class. This is a very rigid macrocycle here, porphyrin chromophore. So if it aggregates, they mostly show quenching. So that is sort of a preconceived, I would say, in some sense.
So if we introduce a benzene linker in between both, the rotor will be formed then in the porphyrin molecule. I'm
Sorry,
just saying,
once
I'm
again, you
seeing a
can tell.
linker between the ring and the porphyrin ring. If there is a linker, a benzene ring is introduced, will
Uh-huh.
it form a molecular rotor?
See, that needs to be established. I mean, possibly one can actually engineer those molecules and look at it.
Mhm.
If I'm not wrong, maybe a few days back, I was actually seeing some reports coming on porphyrin-based AIGENs, porphyrin-based aggregation-induced
Because
emission form.
they are on
So,
porphyrins. I basically work on porphyrins. That's why I'm asking more on
okay,
the
okay,
things.
okay,
That's why.
okay, okay. Maybe
Okay. Thank you
possible.
very much, sir, for the answer. Thank you.
Sir, I have a couple of questions. First one is,
Yeah.
what should be the structural criteria of the molecule to show a turn-on sensing? You told that it should have some free single bond so that it can rotate around. And apart from that, can any other spectral or structural features give us the turn-on sensing, or only
Okay.
that much is required?
So, can you repeat the first part?
I was telling that you told that the free rotation is one the structural features that can
Yeah.
be used in developing the turn-on sensing. But
Yeah.
apart
So,
from
so
that,
ideally, turn on emission, if you want to see, in any case, let us say, whatever be the mechanism, you want the molecule to be very weakly emissive or non-emissive in its free form.
yeah,
Okay. So that mechanism can be anything. So ideally, what we have chosen is that this molecule should have a lot of flexibility in its structure, right? Now, okay, so a lot of flexibility in the structure should be there. So that is how a chromophore, which is weakly emissive in the free form, and for that weakly emissive nature, molecular rotor nature is required. Now, to answer your question, is there anything else? Let's suppose take pyrene. That's another example. Pyrene is not molecular rotor. Pyrene is not molecular rotor. What we can do, pyrene we can functionalize with a cationic group. Let us say an ammonium group or a tetramethyl ammonium group with pyrene. Now that will also come and bind. Now what will happen to the pyrene? It is very well known that if pyrene aggregates, what we found is an excimer. What we find is an excimer. So the idea is to choose a class of molecule which upon aggregation in the presence of heparin should display distinct changes in its photophysical properties. So that distinct changes can be through excimer also. So people have done with the pyrene also. So pyrene they functionalize with a cationic group so that it goes and binds to the heparin, and since multiples of them can bind, so you will have excimer emission. So not necessarily that you have to choose a molecule which has to be of molecular rotor nature. But since molecular rotors are very weakly emissive in the ground in its free form, so once it aggregates, you will see boosted emission, large emission, because pyrene, what happens? Pyrene, you see monomer band decreases, and there's a very slight change in the emission, but new band comes up. So pyrene is more advantageous in terms of ratiometric response. So there are logics for choosing molecules, and the first logic is we should be cationic, and upon aggregation it should have a large change in its photophysical properties like absorption and emission features. For our purpose, we have chosen molecules which are of molecular rotor nature, but for the same reason somebody can take actually a pyrene derivative or an anthracene derivative for that matter. But you will have to ensure that once that aggregate, you will have a new and distinct properties. So that's how it has to be chosen.
okay, sir. And what about the J aggregate and H aggregate, which causes the blue shift and red shift in the spectra, sir? How we can correlate that?
Correlation in terms of you are telling spectroscopic properties wise, or you are telling in response of heparin sensing?
Spectroscopic properties.
Yeah, so so there's a very standard, it's a textbook material these days. So you have this exciton theory by Casa's exciton theory. So what happens that when the two molecules are in the aggregated form, so in the excited state there is a splitting. We say excitonic states. So you have a common ground state, but you have two excited states splitted, right? Because of the excitonic interaction. Now spectroscopically, what happens is that, so there are certain selection rules also. So what happens is that for the H aggregate, so again I'm telling, so if you can follow, there are two excited state and one ground state. Now in the case of H aggregate, the only the transition to the higher excited state is allowed, not the lower one. Okay, so since if it is higher, more energy is involved, so then it is blue shifted. Whereas in the case of J aggregate, the transition to the lower excitonic state is allowed. So depending on the geometry of the aggregate, the selection rules are defined. In the case of H aggregate, transition to higher energy level is allowed, and in the case of J aggregate, transition to lower energy level is allowed. So lower energy means lower energy gap, so you get a red shift, and in the case of higher excitonic state, more energy is involved, so you get a blue shift.
Sir, one last question. Can we increase the selectivity by changing the pH of the medium?
Well, I think it depends on the probe. In some cases it may be, but in our case I would say, suppose if I change the pH of the medium. See, in most of the cases you might have seen I have taken groups where the pH cannot change the state of the molecule. If you take an ammonium group as a cationic group, so then when you lower down the pH, that ammonium will get protonated, NH4 plus. If you come to higher pH, then it will become neutral, the charge is lost, right? But in my probes, I have taken groups where the charged state of the probe molecule is not sensitive to the pH of the medium. Now what will happen to heparin? If I change the pH of the medium, if you see the heparin structure, there are 3 sulfonate groups and 1 carboxylic acid group. So what might happen at a lower pH, because sulfonates are highly ionized, at any pH it will remain ionized state. But carboxylic acid group might get neutralized at the lower pH. So if you are talking about selectivity, then it is from the perspective of the probe, the optical probe. And in that case, one can actually choose a probe molecule whose protonation state can be changed with the pH. If you take ammonium, so where you can play with the proton going and coming back. But in most of my cases, I have taken methyl pyridinium group kind of, or methyl ammonium kind of group, where actually protons should not be able to play a role. But on extreme pH, if you go on other side, like going below pH 3 or beyond, then you have to understand that you are increasing the ionic strength of the medium, more H plus, so ionic strength changes, and that affects the stability. Yeah. So in a nutshell, to answer your question, at least with the probes that we have chosen, selectivity cannot be improved by tuning the pH of the medium.
Yeah. Okay. Thank you, sir.
Thank you very much, sir, for the insightful lecture. We are very delightful to welcome you here. And in the future also, we want you to come offline in the campus and just deliver us a talk.
Yeah, whenever it is possible, if I happen to travel that side, I mean, definitely will try to interact with you guys.
Thank you, sir.
So now I thank Dr. Ritika Madam to chair this session, and I also thank Dr. Prabhat Singh for giving an insight into the application of absorption and fluorescence spectroscopy. We'll be delighted to have such more talks in near future, and we'll definitely be in touch. So now here we'll end up with the second session of the first day of the FDP. So from tomorrow we'll start from 10:30. Today we had some delay because of some other involvement of the speaker. So from tomorrow we'll have the third session, that is on IR and rotational spectroscopy. So I request you all to join again tomorrow. Thank you. So here we'll end up the second section of the FDP.
Thank you so much for having me here. Okay. Bye bye.